This is the lecture for Chapter 5, Microbial Metabolism, Part 2. In Part 1 of this lecture, I went over the fact that cells need to be able to control their biochemical reactions. And to do this, they use biological catalysts called enzymes. And enzymes are composed of two major parts, an apoenzyme, which is the protein part of the enzyme, and then they also must have a coenzyme or a cofactor. And the differences between the coenzyme and cofactor is that a coenzyme is an organic molecule, so a molecule that contains a lot of carbon, high percentage of carbon, and a cofactor is an inorganic molecule, so that could be an ion or a metal. The apoenzyme will bind to a coenzyme or cofactor to form the holoenzyme. And the holoenzyme is the whole enzyme, the active form of the enzyme. And every holoenzyme has a particular active site, which will fit perfectly the shape of the substrate the enzyme binds to. And both the apoenzyme and the coenzyme or cofactor are needed to make up a complete active site. The process by which enzymes function is summarized in this figure. The holoenzyme has an active site that is specific for one particular substrate. And the enzyme will bind that substrate in the active site, and in doing so will form an enzyme substrate complex. And it's during the formation of this enzyme substrate complex that induced fit will occur. An induced fit is where the enzyme and substrate will form hydrogen bonds between the two. So everywhere the enzyme and the substrate interact, that will form hydrogen bonds. Those hydrogen bonds will put stress on the covalent bonds in the substrate, causing them to break or reform, and that will create the products. And because the products are not the same shape as the substrate, they will be released. And then the enzyme, which is unaltered by the whole process, is ready to do the reaction again. Enzymes are proteins, and protein structure is very important to how enzymes function and to their specificity. So when we talk about proteins, when we talk about their function, we say their function equals their structure. And we say this because any change in structure will change the function. And this is true about enzymes. Any change in their structure will change their function. Also, protein structure relates to the specificity of enzymes. Every enzyme will recognize and bind to only one type of substrate. And also, each enzyme will catalyze only one type of reaction. So if you have an enzyme that recognizes starch and can break down starch, causes a catabolic reaction to starch, then that enzyme will not be able to add glucose to starch. It would not be able to catalyze an anabolic reaction with starch. So each enzyme will bind only one type of substrate and catalyze one type of reaction. Protein structure is determined on a lot of different levels, and it begins on the genetic level. So if you remember from an earlier chapter, a gene is a sequence of DNA that determines the structure of a protein. It's going to determine the primary structure of a protein. So a gene is the term that is used for a sequence of DNA that can be transcribed into a messenger RNA, and that messenger RNA is then translated into a sequence of amino acids. And that initial sequence of amino acids is considered the primary structure of a protein. So again, the primary structure is that sequence of amino acids. Secondary structure is how that long chain of amino acids interacts with each other by hydrogen bonding to either form an alpha helix or a beta pleated sheet. Tertiary structure, tertiary 3, this is the three-dimensional structure of the protein. That's how the whole thing winds up upon itself to form a polypeptide. And then quaternary structure is how that polypeptide interacts with other polypeptides to form a functional protein. So things that can alter protein structure. Changing the sequence of the DNA template can change the structure of a protein. If, for instance, there is a mutation and this adenine is converted to a cytosine, then that will be transcribed into a guanine at this codon. So then this codon becomes GUU, which will no longer be phenylalanine. It will now be valine. So this, a mutation in the DNA, can cause a change in the primary structure of the protein. And depending where this mutation occurs, where this missense mutation occurs, that can cause a change in the secondary structure, which will lead to a change in the tertiary structure and a change in the quaternary structure. And usually if there's a change in structure, there's a change in function, and for the proteins, they don't function well. Another way to change protein structure is to have an effect on hydrogen bonding. So if you change the ability of the amino acids to hydrogen bond with each other, you're going to change the secondary structure, which could lead to a change in the three-dimensional structure and so on. Or changing the hydrogen bonding could change the tertiary structure, which could change the function of the protein. So some of the major ways to change protein structure is to mutate the DNA or to have an environmental effect that alters the hydrogen bonding within the protein's three-dimensional shape. When a protein has lost its tertiary structure, we say it has denatured. So here is an active protein. This is an active protein, and this is an example of what its tertiary structure would be. And you can see some characteristics from secondary level of structure, alpha helices, and a beta pleated sheet there. So this is the active or functional structure, three-dimensional structure of this protein. And if it is denatured, then it loses all of that structure. And it still has its primary structure. It still would have its sequence of amino acids, but all of that three-dimensional structure is gone. And that is usually due to a loss of the hydrogen bonding, which was holding it together. There are several different factors that can affect enzyme activity in a cell. And the very first factor is synthesis. This is referring to whether the enzymes are actually being produced or not. So another way to look at this is gene expression. So are the genes being transcribed and translated so that the enzymes are produced? In eukaryotic cells, there are many levels where gene expression is controlled. But in prokaryotic cells, there's only one way to control gene expression, and that involves operons. This is the structure of an operon, and remember an operon is a segment of DNA. And the structure of the operon, it has a regulatory gene, downstream is a promoter, an operator, structural genes, and then at the very end would be the terminator. And remember that operons control gene expression at the transcriptional level. So for transcription, you need an RNA polymerase to recognize the promoter. So this would be the RNA polymerase. The RNA polymerase would recognize the promoter. It would bind to the DNA at the promoter. Then it would slide along the DNA, transcribing everything between the promoter and the terminator. Once it gets to the terminator, then it would release from the DNA. So that's where the transcript comes from the RNA polymerase transcribing the DNA from between the promoter and the terminator. And then of course that messenger RNA would then be translated, and then you get the production of those enzymes. Operons control transcription by using the product of the regulatory gene, which is the repressor. And the way this is controlled is that the repressor can recognize the operator. If the repressor is active and binds to the operator, then that blocks the RNA polymerase from sliding along the DNA. And if the RNA polymerase cannot slide along the DNA, then there's no transcription. If there's no transcription, there's no mRNA, there's nothing to translate, and no proteins are produced. So the first way to control enzyme activity in prokaryotic cells is by using an operon and controlling the gene expression of these proteins, of these enzymes at the transcriptional level. Another factor that affects enzyme activity is temperature. This is a graph that shows the relationship between changing temperature and the effect on the enzymatic activity. The x-axis is temperature in Celsius, the y-axis is enzymatic activity. Every enzyme has an optimal temperature where it functions at its highest rate. And if you change the temperature from that optimal temperature, you will get a decrease in activity. And it doesn't matter if you take that optimal temperature and you increase temperature, you will get a steep decline in activity, whereas if you decrease temperature, you will get a gradual decline in activity. And actually what that is indicating is that it's showing you that the changes in temperature are going to have different effects on the protein. When you increase temperature, what is happening is that the protein is being denatured. So as you increase temperature, you start to disrupt the hydrogen bonds, which will denature the protein. If you warm up the temperature just a little bit and then go back to the optimal temperature, the protein can renature. It can go back to its three-dimensional shape. But if you heat it too high, then you'll get permanent denaturation and you cannot go back to that natural shape. And if you cook food, you know that this happens to protein that you cook. For example, an egg. The whites of an egg, or egg whites, are composed of protein called albumin. And the normal shape of the albumin creates something that we can see as a clear, sort of viscous kind of substance. Now, if we apply heat to this egg, if you apply a little bit of heat to the egg, the protein will denature a little bit, and then if you cool it down, it will go back to the original shape. But if you apply a lot of heat to the egg white, the albumin, will become permanently denatured, and it will have a completely different look. It will be more solid and opaque. So that's what happens when you add heat to a protein. A slight increase in heat can be reversible, but if you have a big increase in heat, a significant increase in heat, you are going to permanently denature the albumin. And you should know that there is no way to get it back to the original form once it has permanently denatured. Cooling, reducing temperatures, has a different effect. And when you reduce temperatures, what you are doing is reducing the ability of the enzyme to perform induced fit. Induced fit. So the enzyme is not denaturing, you're just slowing down its ability to perform induced fit. And that, of course, means being able to form hydrogen bonds with the substrate to stress the covalent bonds within the substrate to form the product. An example of this is you can take the albumin, you could take egg whites, and you can freeze them actually. So if you have leftover egg whites from a recipe, you can actually take those egg whites and freeze them. And then when you freeze them, of course, if these were enzymes, they couldn't function. But if you warm them up, they will actually go back to the original shape. And that's the same with enzymes. So with enzymes, if you cool them down, they do not denature. They just don't function because they don't have the ability to perform induced fit. But if you warm them back up, They will go back to their original shape and they can function again. So changing temperature will have two different effects on a protein or on an enzyme. Every enzyme has an optimal temperature. If you increase temperature, you are going to denature the enzyme, possibly permanently, and it will be unable to function again. If you reduce temperature, you're going to have an effect on induced fit. So if you reverse that and warm it back up to its optimal temperature again, it will be able to function again. Another factor that can have an effect on enzyme activity is pH. This is the graph that shows the relationship between changing pH and the level of enzymatic activity. Just like temperature, every enzyme has an optimal pH where it functions at its highest rate. And as you increase pH, you'll get a decrease in activity from the optimal. And as you decrease pH, you'll get a decrease in activity from the optimal. And if you look at the curve, what you will notice is that both sides of the curve are symmetrical. So this is telling you that changing pH by either increasing or decreasing is going to have the same effect on the protein, same effect on the enzyme. And that effect is going to be to have an effect on the hydrogen bonding. So remember, pH stands for the measure of relative concentration of hydrogen ions in a solution, and the relative concentration of hydrogen ions in a solution is going to have an effect on hydrogen bonding. And hydrogen bonding is very important for induced fit and also holding the three-dimensional structure of the enzyme. So changing how hydrogen bonding occurs is going to change the secondary and tertiary structure of the enzyme. And if you have an extreme change in pH, the proteins will denature permanently. An example of this is using food again. So shrimp, this is a picture of raw shrimp, and if you've seen raw shrimp, you know the color is kind of a bluish or a grayish color, and the meat looks kind of translucent. And you should know when you cook the shrimp, it becomes more solid, white, and pink. So that's an indication when you apply heat to the shrimp that the shrimp has cooked. Well, another way to sort of cook shrimp is to mix the shrimp with an acid, like lemon juice or lime juice. And when you do that, the shrimp ends up looking as if it is cooked. That's because the acid is denaturing the proteins the same way heat would. So I've heard some people refer to this as that the acid, the lemon juice, cooks the shrimp. Well, of course, it's not cooking, it's not applying heat, but it is denaturing the proteins exactly in the same way that increasing temperatures would. So decreasing pH will denature proteins, increasing pH will do the same thing. Another factor that can have an effect on enzyme activity is substrate concentration. And this is the graph that goes over that relationship. As substrate concentration increases, enzymatic activity will also increase to a point. And it will get to a point that's called the saturation point. And the saturation point is basically the fastest rate that the enzyme can function. So even though you keep adding more and more substrate, the enzyme has a limited rate at which it can react. And this is another depiction of it. With low substrate, you have the enzymes. They'll be able to catalyze all the reactions fairly quickly. As you increase the substrate, you get to a high level of substrate. The enzymes are functioning as fast as they can, but they have a limit, and that is called the saturation point. The last factor that affects enzyme activity that I want to discuss are inhibitors. And inhibitors are molecules that bind to enzymes and stop them from functioning. So an example of a normal holoenzyme is here. This is the holoenzyme. It's composed of an apoenzyme bound to a cofactor or a coenzyme. And together, they form an active site in the holoenzyme, and that active site fits perfectly the substrate that the enzyme is specific for. So the enzyme will bind the substrate, perform induced fit, which will stress bonds, covalent bonds in the substrate, converting it to the product. A competitive inhibitor is a molecule that competes with the substrate for the active site. So a competitive inhibitor is a molecule that actually binds to part of the active site and prevents the substrate from binding. So that's a competitive inhibitor. So it does not change the shape of the enzyme in any way. It just prevents the enzyme from binding to the substrate. A second type of inhibitor is a non-competitive inhibitor. And a non-competitive inhibitor binds to an allosteric site. Allo means other, steric means form. So this molecule binds to an area that is not the active site, but when the non-competitive inhibitor binds, it changes the shape of the enzyme. And so that changes the shape of the active site, and now the enzyme can no longer bind to the substrate. So that inactivates the enzyme that way. Many inhibitors are considered poisons. So for example, a competitive inhibitor for humans is carbon monoxide. So carbon monoxide will actually bind to hemoglobin instead of oxygen. So that's acting in a way as a competitive inhibitor. A non-competitive inhibitor example is cyanide. And cyanide will bind to the allosteric site of enzymes that are involved in the electron transport chain. And it will prevent those enzymes from functioning. So a lot of inhibitors are actually considered poisons. Because if you stop the enzymes from functioning, you'll stop the cell from being able to perform biochemical reactions, which will most likely lead to the death of the cell. Most cells, especially prokaryotic cells, do not have unlimited resources. So most cells have to be very efficient with their enzymatic processes in order to produce just enough product at the right time. And feedback inhibition is one way for cells to maintain this efficiency. This is a diagram that goes over the process of feedback inhibition, but I also want to say a basic definition. Feedback inhibition is where the end product of a series of enzymes acts as a non-competitive inhibitor on the first enzyme. So now I'll go through the process. First thing to note is that most reactions actually involve more than one enzyme. A lot of basic biology teachers, and even I, may have misled you a little bit in giving you the impression that one enzyme catalyzes one whole reaction. In reality, each enzyme is so highly specific that each enzyme only does a part of a reaction. So in this example, we start out with a substrate, and the substrate is going to be converted to that end product. And in this example, it's going to take 3 enzymes to do it. Enzyme 1, enzyme 2, and enzyme 3. So this is a series of enzymes. So enzyme 1 is going to bind to the substrate, convert it to an intermediate. Then enzyme 2 is going to bind to that intermediate, convert it to a second intermediate. And enzyme 3 is going to bind that second intermediate and convert it to the end product. So again, most enzymes are so highly specific that they only catalyze part of a reaction. So this is the series of enzymes, enzyme 1, 2, and 3. So the end product is whatever that little end product is. Now, if for whatever reason the cell is not using that end product and the concentration of that end product is increasing and increasing, then to shut down this process to be efficient and not make too much end product, that end product of a series of enzymes is going to act as a non-competitive inhibitor. So it's going to bind to the allosteric site of the first enzyme. And when it does bind to the allosteric site of the first enzyme, it's going to change the shape of the enzyme so it can no longer bind to the substrate. And that's going to shut down the pathway. Because if enzyme number 1 does not catalyze its reaction, then there's nothing for enzyme 2 to work on. And if enzyme 2 has no reaction to catalyze, there's nothing for enzyme number 3 to work on, and you stop making that end product. So again, the definition of feedback inhibition. Feedback inhibition is where the end product of a series of enzymes acts as a non-competitive inhibitor on the first enzyme. For the rest of the lecture, I want to go over the different types of carbohydrate metabolism. And carbohydrate metabolism is very important for all cells because this is how cells take glucose and they break it down to form ATP. And ATP is the readily usable form of energy that all vegetative cells need in order to function. Aerobic respiration is one of the pathways that cells can use to metabolize glucose and produce ATP. And probably the first time you learned this, it was referred to as cellular respiration. And this would have been similar to the diagram you would have seen then. Aerobic respiration involves 3 processes: glycolysis, citric acid cycle, which was referred to as the Krebs cycle, and the electron transport chain. And when you first learned this as cellular respiration, you probably learned about it in terms of a eukaryotic cell. And of course, this process occurs in the mitochondria. So glycolysis would have occurred in the cytoplasm, and then the citric acid cycle and the electron transport chain were in the mitochondria. This process can also occur in bacteria. So this is a diagram of a bacterial cell, and this blue is the cell membrane, and this would be the inside of the cell, and this is the outside of the cell. And when a bacterial cell performs aerobic respiration, it also goes through glycolysis and the Krebs cycle, or the citric acid cycle, and then those electron carriers go to the electron transport chain, which is actually embedded in the membrane. Electron transport chain embedded in the membrane of the bacterial cell. But both processes, whether it's in a eukaryotic cell or a prokaryotic cell, are similar. Now I'm going to go through the steps of aerobic respiration. And the first step is glycolysis. And glycolysis, glyco refers to glucose and lysis is to split. So in this process, the glucose molecule is basically split in half. And while that sounds very simple, the actual process takes 10 steps to go from 1 glucose molecule to 2 pyruvate molecules. So it's an involved process. And with this, you don't have to know the individual steps, but you should know that in order to split the glucose, it involves an input of ATP, 2 ATP molecules. And once it is split, it will generate 4 ATP molecules. But then that's only a net gain of 2 because it cost you 2 initially. But also it's going to generate some electron carriers. So that's NADH. So glycolysis is basically the process of splitting glucose from 1 molecule into 2 pyruvate molecules, and it will generate a net gain of 2 ATP and also 2 electron carriers, NADH. The second stage of aerobic respiration is the citric acid cycle, or formerly known as the Krebs cycle. And this is a diagram of those reactions. And for this, the pyruvate that was generated during glycolysis is going to be converted into acetyl-CoA so it can enter into the cycle. And during the process, that acetyl-CoA is going to be oxidized. And oxidized means that the hydrogens, the energy is going to be removed from that molecule. And in doing so, it's going to generate a little bit of ATP, but it will also generate electron carriers, NADH, and will also generate another electron carrier, FADH2. And also during this process, this is where the waste product of carbon dioxide is produced. So for the citric acid cycle, pyruvate needs to be converted to acetyl-CoA. Then as that acetyl-CoA is oxidized, it's going to generate a little bit of ATP, electron carriers, NADH and FADH2, and the waste product of CO2. The last stage of aerobic respiration is the electron transport chain. And the electron transport chain is very different compared to glycolysis and the Krebs cycle because the electron transport chain is a physical structure and it is a process. Glycolysis and the Krebs cycle, that involved a series of biochemical reactions. The electron transport chain is very different. So this is the structure of the electron transport chain, and this requires proteins, so protein complex, to be embedded in a membrane. And these proteins are redox proteins. So redox proteins, that means that they take electrons and they pass them from protein to protein to protein. And so that is why they call this the electron transport chain, because the electrons are going to flow along these proteins. And that electron flow is going to power some of the proteins that are called proton pumps. And they're called proton pumps because they take the protons, which are also known as the hydrogen ions, and pump them to one side of the membrane. So on this side of the membrane, there is going to build up a high concentration of hydrogen ions. So that is going to build up a high concentration of hydrogen ions on this side, and the other side of the membrane is going to have a low concentration of hydrogen ions. So that creates a concentration gradient of hydrogen ions, and then the ATP synthase is going to allow those hydrogen ions to diffuse back to the area of low concentration. And in doing so, the movement of these hydrogen ions flowing through the ATP synthase is going to power the production of ATP. So that is how the electron transport chain functions. All these electron carriers, the NADH, the FADH2, are going to drop their electrons off at the electron transport chain at these proteins. The electrons are going to be transported from protein to protein. That movement of electrons is going to power the proton pumps to increase the concentration of hydrogen ions on this side of the membrane so that the ATP synthase can allow them to diffuse back across the membrane, and that flow of hydrogen ions is going to power the production of ATP. Now this whole process is called aerobic respiration. Aero refers to oxygen. And this is considered aerobic respiration because at the very end of this process, the oxygen serves as the final electron acceptor for that electron. So once that electron has passed through the electron transport chain, it needs to go to a final electron acceptor. And that will be the oxygen, and then the oxygen will bind to two protons producing water. So this is the process of aerobic respiration. And a maximum number of ATP that can be created from one glucose is 38 ATP. Again, from one glucose molecule that starts at the very beginning. Anaerobic respiration is the second pathway by which cells can metabolize glucose to produce ATP. And ana means without, aero means oxygen. So this is a type of glucose metabolism that does not involve oxygen. But it is called respiration because it is very similar to aerobic respiration. Anaerobic respiration will start with glycolysis, just like aerobic respiration. So remember, glycolysis involves taking glucose and breaking it in half to form two pyruvic acid molecules. And in doing so, generate some electron carriers and also a net gain of 2 ATP. Then those pyruvate molecules will go through the process of the citric acid cycle or the Krebs cycle. So the pyruvate will be converted into acetyl-CoA. It will run through the citric acid cycle, and in doing so, it will generate a little bit of ATP and also more electron carriers, NADH and FADH2, and also the waste product of carbon dioxide. The last stage of anaerobic respiration will also be the electron transport chain. So all those electron carriers, NADH, FADH2, will carry those electrons down to the electron transport chain. Those electrons will flow along the proteins, powering the proton pumps to pump protons out into one section of the cell, building up a high concentration, and then those protons will be allowed to diffuse through the ATP synthase to generate ATP. So until this point, the process is the same. The difference comes in the final electron acceptor. Since this is anaerobic respiration, the final electron acceptor is not oxygen. Instead, it is some other inorganic molecule. So that could be a nitrogen molecule, it could be a phosphorus molecule, it could be a sulfur molecule, but it is not oxygen and it is not organic. So anaerobic respiration is almost identical to aerobic respiration with the exception of that final electron acceptor. And the maximum number of ATP that can be synthesized during anaerobic respiration is slightly less than aerobic respiration, and it would be a maximum of about 36 ATP. The third and final carbohydrate pathway is fermentation. And fermentation is a lot easier than aerobic respiration and anaerobic respiration because fermentation is just glycolysis. And in glycolysis, remember, a glucose molecule is split to form two pyruvic acid molecules. And in this process, you will generate 2 ATP. So every one glucose molecule will generate only 2 ATP. But in addition to those 2 ATP, there are 2 electron carriers generated. So there will have to be a final electron acceptor in fermentation also. And in fermentation, the final electron acceptor is some sort of organic molecule. There are many different types of fermentation, and they all have different final electron acceptors. The two that you might be most familiar with are lactic acid fermentation, where lactic acid is the final electron acceptor, and it's an organic molecule, and also alcohol fermentation, where ethanol is the final electron acceptor, and that is an organic molecule also. Another difference between lactic acid fermentation and alcohol fermentation is that with alcohol fermentation, there is production of 2 molecules of carbon dioxide. With lactic acid fermentation, there's no production of carbon dioxide. With fermentation, there are many different types. And you should just be aware that there's more than just the simple lactic acid and alcohol fermentation, but you don't have to memorize them. But with all of these, the fermentation end products, these are the organic final electron acceptors. This table compares and contrasts the 3 types of carbohydrate metabolism. So the 3 types are aerobic respiration, anaerobic respiration, and fermentation. And aerobic respiration is the only one that requires oxygen to be present. In terms of final electron acceptors, aerobic respiration uses oxygen. Anaerobic respiration uses some other inorganic final electron acceptor. and fermentation uses an organic final electron acceptor. And the last important thing is the relative number of ATP that is produced from one glucose molecule. Aerobic respiration, you can get a maximum of 38 ATP in prokaryotes. Anaerobic respiration is less than 38 ATP, and fermentation is only 2 ATP from one glucose molecule. Some organisms can perform both an aerobic respiration and another anaerobic pathway, and the usual pairing is aerobic respiration and fermentation. So a lot of bacteria that can perform aerobic respiration have the ability to use a metabolic pathway that does not include oxygen, and if they do, it's fermentation. Human cells have the same thing. We perform aerobic respiration, and our cells also have the ability to perform fermentation. It is very important for vegetative cells to have a constant source of energy. And often the preferred source are carbohydrates, where the carbohydrates will go through a process of carbohydrate metabolism, either aerobic respiration, anaerobic respiration, or fermentation, to produce the ATP that the cell needs. But if the cells do not have a source of carbohydrates, instead they can use lipids. And often the role of lipids in living cells is to be the long-term storage for energy. So if the cell runs out of carbohydrates, they can use the lipids, and the lipids can enter into this metabolic pathway in a couple different points. If the cell does not have a source of carbohydrates and does not have a source of lipids, then it can use proteins also to produce energy. So those amino acids can enter at several different points in this pathway. But the point of this is that production of ATP is very important to the cells, and they have several options to use to generate ATP. This idea of alternate biochemical pathways also leads us back to the idea of metabolism in general. Originally, I showed you this figure of all the known metabolic pathways in a cell. And here is the carbohydrate metabolism with glycolysis here, and then the Krebs cycle here. And of course, this is the general way for cells, most cells, to produce ATP. And so again, if the cell needs ATP and it doesn't have a source of carbohydrates, it can use a source of lipids and filter that through a pathway to enter into ATP production. And also amino acids. If there aren't any lipids or carbohydrates, the amino acids can enter at different points into this pathway. All of those are catabolic reactions. But even the idea of alternate pathways relates to anabolic reactions. So if the cell needs to build up proteins and it needs to synthesize more amino acids, it can take carbohydrates and go different pathways to generate those amino acids. If the cell is going to replicate its DNA and it needs to create more nucleotides, it can take lipids, it can take carbohydrates, follow different pathways using different enzymes to generate those nucleotides it needs. So the cell is constantly monitoring what catabolic reactions it needs to perform and what anabolic reactions it needs to perform. And the enzymes are vital for all of these reactions. If the cell's enzymes can't function, then the cell can't metabolize. And if the cell can't metabolize, the cell can't function and the cell will die.